today to talk to you about your science fair project and how you can make it more scientific. So first, let's talk about why you're listening. I'm sure, of course, that you'd like to get a good grade on your project. And we'll also help to give you some tips about your science fair project, and you'll also see a judge's perspective on science fair projects. We'll go more into the scientific method too, so you can learn more about that. So we wanted to introduce ourselves before we go any further. I'm Casey Lozier, and I have a biology chemistry degree from Manchester University. Pearl Feaster and Paul Sustek both have science degrees from IPFW, and we're all second-year Manchester University College of Pharmacy students. And as you can see here, we've all judged the science fair here in Fort Wayne a few times. So what are we going to be doing today? Well, first we'll tell you what the judges are looking for in your project. Then we'll go over the scientific method and give you some examples about a science fair project. So this is taken directly off the science fair website, and it gives you a rundown of what the judges look at when they're judging your project. And all of these categories are judged equally, so you want to be sure to work on all of them. The one that was there was the scientific method. So what is it? The scientific method is a process, and it's a way of thinking. And you can use it to learn more about the world. You ask questions about things, and you form a hypothesis to answer those questions. Then you test your hypothesis to see if it's right or wrong, and you collect data through observation and experimentation to test your hypothesis to see if it's true. Some other important components to your experiment should include variables, constants, and controls. Now, variables are anything that changes. Like independent variables are things that you can change on purpose, while dependent variables are what you observe. Now, you change an independent variable to observe the effect it has on dependent variables. Now, constants are anything that stays the same, and they help ensure that you are being consistent in your experiment. An example to explain these concepts better is ice cream making. If you want to figure out how to make the best ice cream, you might change things like the color, flavor, or amount or type of sugar to try to get the best mix. Now, those are your independent variables because you're changing them on purpose. Your independent variables, as I said, the color, flavor, or sugar, will affect how your ice cream tastes and looks. The taste and appearance of your ice cream are the dependent variables that you will be measuring. To ensure consistency, you will make sure that all your batches of ice cream use the same amount of milk and ice. Those are your constants, and they help ensure that all the changes in your ice cream are just based on the things that you want to change, like the color, flavor, and sugar. Your control in this case, which is a standard that you want to compare your results to, will just consist of milk and ice. You'll compare all the batches of ice cream that you make to this standard to see how much better they are, how much worse they are. We also want to explain some common mistakes that we've seen when judging science fair projects, so you can try to avoid them. A big one is students doing demonstrations rather than using the scientific method. You want to make sure that you're finding answers through your experiment. You ask a question and you test your hypothesis rather than just show examples of things that just how things happen. I'm sure you've seen the volcano project where you can see a volcano erupt. While it's certainly cool to see that, it's not really testing anything. You want to make sure that you have a hypothesis to test. Another issue we see is having no controls. You need to be able to compare your results to something. For variables, you want to make sure you don't have too many. You want everything except what you're testing to be similar so that you know that the results of what you find are just based on what you changed. More common mistakes include not applying your background information. You want to use your background information to help form your hypothesis. What does the information that you gathered tell you about the answer to your question? You want to use that information and not just make a random guess on what you think will happen. You also don't want to forget to apply your conclusion. What do your results from your experiment tell you? And lastly, it's important to know everything on your poster. Judges can ask anything about it, and you want to make sure that you're prepared and can have answers to everything.
Now that Casey talked about the scientific method, let's apply the scientific method to a project we came up with to show you how to apply it. This diagram of the scientific method will be at the top of every slide to show you where we are in the process. First, we're going to ask a question. Our question is, what's the best liquid for sprouting seeds? I found out through my background information that plants can grow without soil, and it's called hydroponics. As you can see in this picture, the plant's roots are submerged into the water with nutrients without soil. The water has nutrients such as nitrogen, phosphorus, potassium, sulfur, calcium, and magnesium. Plants that use those nutrients to make energy based on photosynthesis, where carbon dioxide is used to make up plant food and oxygen. Finally, I found out through my research that seeds need water, light, and heat to sprout so that it can become a plant. To find the best liquid for sprouting bean seeds, I wanted to test different liquids. The liquids I chose were apple juice, whole milk, Powerade Zero, and Sprite. I chose all of these for a reason. Based on my background research, I know plants need a certain amount of nutrients, so I picked liquids in my house that have the most nutrients for plants and recorded how much they had. What types of variables are these called? That's right, they're independent variables. Now my original question asked, what's the best liquid for sprouting seeds? Well, what do I mean by best? I decided to measure when seeds would sprout and the length of the sprouts as my way of finding which liquid the seed prefers. What types of variables are these called? Dependent variables. What control will I use to compare my test liquids to? Well, since I know seeds are usually planted in water, this will be my standard, or known as control. My constants would be temperature, light, bean seeds, amount of liquid per test, and water, since all my test liquids have water in them. Based on my test liquids, I decided that my hypothesis is that bean seeds will grow best in whole milk since it has the most potassium and calcium. Next, we're going to test our hypothesis by writing out my procedure, follow our procedure, collect data, and produce results. Before I start the experiment, I'm going to write down my procedure. Basically, I'm going to place 4 bean seeds into each cup and put the same amount of liquid into each cup. I will replace the liquid after 7 days and stop the experiment at 14 days. I will record how many seeds sprouted every day and measure the length of the sprouts every day. What is my control? My control is my standard, where I'm going to compare my independent variables to, so I know plants usually grow best in water. I'm going to use it as my control. As a review, these are my independent variables, or test liquids. I'm going to measure how well seeds grow by when they sprout and the length of their sprouts. So these are my dependent variables. Finally, my constants are things that are the same for each cup, which is that they all have bean seeds, they all have 4 seeds, they all have the same amount of liquids, they all have fresh liquid in 7 days, and they will all grow at the same temperature. Now that I know my procedure, I'm going to record data every day by counting how many seeds sprouted and measuring the length of their sprouts. What types of variables are these called? You know it. These are my dependent variables in which I'm measuring. Now that I did my experiment for 14 days, I want to present my results. I decided to keep the number of seeds sprouted every day in a table format. As you can see, the seeds in Powerade sprouted first, but water sprouted the fastest among all the seeds since it had the most number of days with all 4 seeds sprouted. So as you can see, there's more 4s here. Milk has no sprouted seeds. My general statement on these results, I would say, is water is the best, then Powerade, apple juice, Sprite, then milk is last. Next, I created a chart that's easy to compare the length of the sprouts. As you can see, none of the sprouts of the test liquids had lines similar or higher than water. So I can see this is water, and none of these lines of the other liquids were close or even better than water. My general statement on this chart is that water had the longest sprout length than apple juice, Sprite and Powerade were similar, and milk didn't grow at all. Now that I understand my results, let's go back to our testable hypothesis. My hypothesis was that bean sprouts will grow best in milk, since it had the most potassium and calcium than all the other liquids. Now let's come up with our conclusion and figure out what this means. First, our hypothesis was not correct. It was clear that bean seeds grew best in water because the seeds in the other liquids did not have higher or similar numbers of sprouting seeds and average sprout length. This is a good conclusion, but let's not stop here. What does my conclusion mean? Why did my bean seeds grow best in water? Could we explain this? Based on my background information, plants require light to grow, and milk is not a clear liquid like all the other liquids. Also, apple juice and Sprite have a high sugar content, which cause the seeds not to grow as well. Basically, our conclusion for this experiment is do not use our tested liquids for hydroponics. What are some of the limitations to our experiment? First, our experiment was only 14 days long, and so our results could have changed if it was longer than 14 days. So in other words, maybe apple juice would have actually been higher than water if we would have done the experiment longer. Also, none of our liquids had nitrogen and phosphorus, which are required for plants. Therefore, plants do not drink the same drinks as humans. If I were to do this experiment again, I would include liquid, nitrogen, and phosphorus and find which best nutrients for plants and continue this experiment past 14 days.
Thank you, Pearl. So now we will go over an example of an experiment that we saw at last year's Regional Science Fair. We will analyze it and try to identify things that the student had done well and also things the student can improve upon based on all the things Casey and Pearl have gone over so far. We will do this so hopefully you will be able to take away some good ideas for your experiment. We first start out, as Pearl said, by asking the question. The student in this example asked the question, out of the 4 common ways of boiling water, which method is the fastest? What do you think of this question? Well, there are some good things about this question. For instance, the student defines that they would boil 2 cups of water, which will be a constant for them throughout all of their trials. And they will measure which boils the fastest, which is easily measurable with a clock or stopwatch. Now, there are also some problems with the question as well. The student refers to common ways of boiling water. That can be a vague statement because what seems common to the student in this experiment may not be common to you or me. In your experiment, you will want to be more specific. The other problem is figuring out exactly when water starts boiling can be a challenge. If you have ever boiled water or helped your mom boil water to cook noodles, you may have noticed that when the water is starting to get very hot, it starts out with 1 or 2 bubbles at first, and then more and more bubbles, and then finally it's boiling. It becomes a rolling boil. The student in this example will need to define exactly what boiling is so that they stop their stopwatch at the same time for every trial of their experiment. Otherwise, it will add an unwanted variable to their experiment. This slide is about gathering background information. At the student's house, they will be boiling water in a copper kettle, 2 different sizes of steel pots, and a glass bowl in the microwave. So to begin, the student searched online or in the library information on things like water, heat, steel, copper, and microwaves. The student then reported some facts about each, which you can see on the slide. The main one was that copper is very conductive compared to steel, which is very important. They even found out that microwaving water works a lot differently than on a stovetop. However, even though the student found out that information about the differences between microwaves and stoves, they still continue to test the glass bowl in the microwave, even though it is not the same. This will make it more difficult for the student to keep their comparisons constant throughout all of their different trials. So then, based on the background research, they made their hypothesis that the copper kettle should boil water the fastest since it's the most conductive. They will compare the kettle with the 3 other methods mentioned earlier. So they did a good job of using their background information here to lead them to their hypothesis, which they will now test. The procedure for how you will carry out all of the trial tests needs to be laid out ahead of time so that it is the same for all of the trials. For this student's experiment, they lay out that they will carefully measure 2 cups of water and add it to the container which they're testing. The 2 cups of water is one of their constants. Then they will turn on the gas stove, or in the case of the glass bowl, the microwave, and start the timer at the exact same time. And then when the water bubbles, they will stop the timer and record the time on a chart. So the student did a good job here defining how each of the trials will be performed so that it remains constant. It is also important that you repeat the experiment for every container. The student does this, as they mentioned, at the end of their procedure, and it will help the data to be more consistent so that if there are any mistakes or fluctuations, it can be averaged out in the data collection portion. The two things they maybe needed to do a better job of is, like I mentioned earlier, define exactly when you will stop the stopwatch to measure when it starts boiling. Is it on the first bubble, or is it when it's in a rolling boil? The data collection portion is where you collect and organize all the data. We see on this slide that the top picture is a graph of all the data collected in the 4 trial runs of each container. On the bottom graph, they show an average of the 4 containers based on their time results. It is important that in whatever experiment you choose, you take averages for repeated data trials. That way it will be the most stable data, and it will eliminate any outliers or mistakes from your data. Finally, the results section is where you further organize your data into a nice and neat table where it can be easily seen the results that you came up with. The student in this example did well to organize it and display the data that we could all see. And the next is the conclusion. You're to draw the conclusion based on all the data that you collected from the experiment. This student found that the copper kettle was the fastest out of the stovetop containers, but overall, water boiled the fastest in the microwave in the glass bowl. These conclusions are correct based on the data they collected. Like I was saying before, though, it is hard to compare the stovetop boiling systems with the microwave ones. Another thing that you should think about, as Pearl briefly went over, is to think about things you could do differently or things you would change if you were to do the experiment again. This student said that they would maybe try an electric stove instead of a gas one for their stovetop experiments, or they would use the same container for all the trials and add different amounts of water. So now that we have gone over the whole experiment, what do you think of it overall? Based on their experiment, can we really say that microwaves are the fastest way to boil water? Well, from our determination, we thought that there were too many variables that were not accounted for and not enough constants. The only constant that we saw in the experiment design was that the student used 2 cups of water for boiling in every trial. The variables, on the other hand, were numerous. There were many different container sizes, as you saw in the picture of their kettles and pans, and different energy sources used, like I said, the gas stove versus the one in the microwave. Beneath that, we listed 3 questions that we thought of while we were reading over the experiment. And these you just want to look at and consider those in reference to this experiment when you're coming up with yours. And you're going to have to be as specific as you can to answer certain questions, and if not, be able to explain them to the judges when you're presenting your poster. So here again, we see the strengths and limitations on the experiment. Think about this example we gave you when you were selecting and working on your project this year. Like I said, the strengths were it had 4 trials for each container, and they used averaging for the data. Some of the limitations, the methods they used were not consistent all the way, and they didn't equally test the container for each method. Like I said, some of them use a gas and others the microwave, and there were just too many variables. So how would you make this project more scientific? If you're testing as your variable, your independent variable, the different container types and material it's made of, like the copper versus the steel, you need to be able to control all of your other variables. For instance, you need to make sure the volumes of the containers is the same, and then also the energy source you use for them, like a stove. And then another thing you can do, like I said, is to define when you're going to stop your timer for the boiling. The easiest thing to do might just be at the first bubble you see, that's when you stop the stopwatch. So this is what we went over today. I hope you were able to take away some good advice from this and be able to apply that to your project. You saw what some of the judges are looking for. We went over the scientific method that you should use on any of your science fair experiments. And then we showed you the two examples. The one that Pearl went over, which is the example with the sprouting beans, and then the example I went over where we looked at a student's project and pointed out some of the good things and bad things for them. And I hope you all will be able to get a good grade based on the presentation we gave today, and just see kind of what judges are looking for your science fair projects. And I hope you learn more about the scientific method, and you'll be able to apply that and have fun when you're working on your project. So that's everything. On behalf of Casey and Pearl, I'm Paul, and good luck on your science fair projects this year.